Hello. So I'm going to introduce Edexcel Biology Scientific article for use with question 8 for Unit 5. This is for the October 2022 exam. I will have full session about this article, which will be 2 hours, and this is a Zoom discussion. So you can find more detail about this program on my website www.tulipdust.com. How it works, for example, it is like this. This is paragraph 5, and I have highlighted in yellow color this sentence. Many laboratories pursued research programs to investigate the roles of these channels in thermal sensation by using genetically manipulated mice that lacked these newly discovered genes. This is the sentence found in the article paragraph 5. Now, I make a possible question based on that sentence. Genetically manipulated mice that lack these newly discovered genes, they are TRPV1 and TRPM8. What does it mean? The mouse with TRPV1 and TRPM8 is called wild type, that is normal mouse. And here, this mouse lack TRPV1 and TRPM8, and this is called knockout models. The question is, explain how this mouse lack TRPV1 and TRP8 gene, paragraph 5. Answer is this. The mutated gene is created for the TRPV1, TRPM8 genes. It doesn't work because it's mutated. And this is coupled to the reporter gene to find expression of the mutated genes. The blue color one is the original gene which is going to silence. After that, in the textbook paragraph to page 294, it's written like this. These mouse are called knockout models. They do this by inserting new genes similar to the gene to be investigated, but which makes the original DNA sequence impossible to read. As a result, original gene is silenced and cannot make a protein. Knockout genes are usually accompanied by marker gene to show that they have been incorporated. So you can see here they do this by uh, inserting a new gene similar to the original gene. This is a new gene and this is the original gene. The new gene is mutated, it doesn't work, and coupled to the reporter gene. So then this new gene, which is mutated one, makes the original DNA sequence impossible to read. So this doesn't read. So then the proteins are not produced from the original gene. So this one should be the answer as it is copied from the textbook. So this is enough to get the marks because the too much detail of this knockdown procedure is not required. But for your interest, I just included a little bit extended version, but you don't want to write it. But for your understanding, it will be important. We take a cell from the blastocyst, and then we add the gene which is mutated to these cells, and after that, these cells place back in the blastocyst. So the blastocyst has mutated gene, blastocyst has normal gene. The red color one, they are the mutated cells, and the blue color one are normal cells. After that, uh, when we have this mouse, we have a normal one. We have mouse express both mutated and normal. We call them chimera. So this chimeric mouse, we're going to cross with the normal mouse. So when we cross with the normal mouse, we can get a version which is heterozygous. That means one is active, the other one is mutated. So if we cross these heterozygous versions, then we get a homozygous recessive, that is a mutated genes. So this never express, they don't express this protein. Okay, this is one example. You will cover 2 hours throughout this research article in the Zoom session. So you can join in the Zoom session and you can find the details from the website www.tulipdust.com.